Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show today, bringing another really awesome guest. Today on the show, we have the honor of being joined by Dr. Carina Kern, the Chief Executive Officer of LinkGevity. They are an AI-powered biotech company. They're driving innovation and drug discovery for both aging and diseases of lost resilience. And Dr. Kern has developed a really fascinating new blueprint theory of aging, which basically takes an integrative approach to understanding aging, combining evolutionary theory, genetics, molecular mechanisms, and medicine, and is used to structure LinkGevity's in-house artificial intelligence systems. Dr. Kern's labs are based at the Babraham Research Campus affiliated with the University of Cambridge, and her research has led to the development of really interesting first-in-class area of therapeutics known as necrosis inhibitors targeting cellular degeneration, and their lead asset is looking to begin phase 2 clinical trials later this year as a potential breakthrough treatment in aging, and has been selected also as one of the only 12 global innovations for NASA's Space Health Program, recognizing the potential to mitigate accelerated aging in astronauts during long-duration space missions, which is a topic we've gotten into previously on the show. I'm excited to get back into it. Dr. Kern has held academic and leadership roles in interdisciplinary networks, including serving as the president of the London Evolutionary Research Network and co-chair of the CleanTech Business Challenge, an innovation co-led by the University College London and London Business School. Dr. Kern did her PhD at UCL Institute of Healthy Aging with a focus on reproductive death associated with aging in C. elegans, which is another really cool topic that we'll be getting into to start things off. But really honored to have her. A lot to get into today. Dr. Carina Kern, thank you so much for taking the time on the show.
Oh, thanks, Ira. It's an absolute delight to be here.
Yeah, it's wonderful having you. I'm really looking forward to a lot of these topics that we get into today. Before we touch on necrosis and necrosis inhibitors and a lot of the places that we're going to be going, I would love to start off with you. November 2020, as I mentioned, you did your PhD, Aging Through Reproductive Death in C. elegans. And, while we're going to be talking about a very unique form of cellular death later on, your work in this area, focused on sort of broader organism death, and some of the very similar concepts in terms of the way not just the system breaks down, let's say, in an organism like C. elegans, repurposing biomass, organ degeneration, but the fact that, you also studied ways to delay, in that case, forms of death from, removing the germline and IGF signaling on that. Love to hear about the beginning and, how this topic interested you. And, I think it's a great place to start with where we're going.
Sure. Let me take a big step back and move you into the 1980s, and to one of the areas where there was quite a boom in the aging space. And the reason for that boom came from this small microscopic model organism called C. elegans. So for those of out there who don't really know what C. elegans is, it's a microscopic worm around the size of a comma after a sentence. And if you've ever come across the Human Genome Project, it was actually first done in this particular model. Why it is so simplistic? The point is, if there is a complex piece of biology, if you study it in this organism, maybe it will give you some insights into the broader complexity of the process in humans. Okay, so in the 1980s, work done identified that if you switch off a few genes, single genes in this organism, you can get lifespan extension up to tenfold. So in humans, that is the equivalent of living to 1,000 years. Fast forward decades later, and well, we just haven't been able to reproduce this in humans. And the point actually isn't that we haven't had enough time to test out these interventions. In fact, they are congenital defects that mimic the same mutations in the worm already present in humans. Laren syndrome is one example. And really, in those individuals, what you don't see is this massive lifespan extension, right? There's small potential effects in type 2 diabetes and cancer, debated, but what you don't get is a vast lifespan extension. So, what my PhD at the time really looked at, and I have to say, really challenged the orthodoxy at the time, was whether or not we could ever translate these single gene effects into humans. And I always look at the PhD as three steps forward, two steps back. And the reason for that is it actually identified that it's possible that the aging process is fundamentally different in C. elegans and these small microscopic organisms compared to humans. And so the point is, aging in humans is not so straightforward. You can't just switch off single genes and see the huge lifespan extension. We have many more diseases. It is far more complex. And of course, at the time, it was saying, well, fine, if not here, it was bittersweet. Well, then where to next? And I
Mm-hmm.
think for me, once I completed the PhD, moving on, to my subsequent career, it was very much the where to next.
Mm-hmm. And one other interesting where to next, and again, before we get to necrosis, a couple of years later was your very interesting paper in Geroscience 2022 with David Gems on, the topic of whether cellular senescence is a misnomer. And, as you're pointing out here, we learned a lot that we couldn't turn off single genes. In sort of this area, you're also challenging, a lot of what's been going on in sort of longevity bioscience the last 10, 15 years. It says senescence is either it's a black or white thing, and there was no shades of gray in between. But as you show in this paper, the way we need to think about things like senescence, and ultimately when we get to necrosis, we'll also be talking about this. There's many varieties of senescence, and as you point out in this paper, very important roles in active remodeling dynamics that go on, wound healing, and regeneration in certain organisms, things like that. Let's talk about this paper for a couple minutes as well, because I think it's a very interesting proxy again to how you think about, the standard, model. That there's a lot of options in between the black and white states of these things.
Absolutely. The aging field is awash with so many different ideas. And I think, again, just taking a step back and looking at these ideas in the bigger picture is something that I frequently do. In terms of the cellular senescence, the simple point there was yes, absolutely. Should we change the terminology? And the issue at the moment is cellular senescence suggests it's always bad, but it isn't. It is a gray area. And there are many useful processes associated with senescent cells. You need it for wound healing, you need it for development. So the question is, why does it fail later in life? But senescence is just one area. You can actually take a step back and go into the hallmarks of aging, a very powerful toolkit in the aging space, right? These are different areas of aging heavily studied. And I think you can look at all of those hallmarks and ask yourself, well, yes, they're very powerful, but where are all of these hallmarks coming from? Why do you see these particular changes later in life? And key is, I'm not the only one questioning it out there, right? I'm not the only one saying we need to dig deeper and ask more questions. But the point is, how do you do that? And really, how do you bridge the gap between these hallmarks of aging, all of these ideas in the longevity space, and then the complexity of medicine, right? And the different medical diseases we see later in life. In fact, if you look at the name of the company, LinkGevity, the name comes from linking the longevity space to medicine.
Absolutely. Absolutely. And that's an extremely important link that, and it will, as we get towards your clinical work, is missing, let's say, quite a bit in this space. And again, I'm glad we're going to be going in that direction in the way you think about the applications of some of these tools. Let's start talking now about cell death and specifically necrosis, because it is a cellular death process state that we don't touch. I mean, we talk a lot about apoptosis on the show, not as much in terms of necrosis, just because it's sort of an afterthought that there's a bunch of stuff that happens and then these cells over here become necrotic, and that's very bad. But we really talk about what's involved in terms, unlike apoptosis, which is programmed, regulated, necrosis is, as you point out, uncontrolled. It's usually due to various stresses or toxins or traumas. Can be very, the cellular and molecular features of it very different, and ultimately the pathologies that we ultimately will target as we get into necrosis inhibitors quite different than what we'd see on the apoptosis side. So if you could take a little time and introduce us to the world of necrosis and all you know about it today and compared to apoptosis, that'd be great.
Yeah, I want to give you a big picture view of necrosis. And of course, I can go into the nitty-gritty, the distinction from programmed cell death and so on. But to give you the big picture view, I now want to move a bit more into this novel theory, the blueprint theory of aging.
Yep.
And remember, I said where to next, right? How do you delve into this complex problem of age-related disease? And I just want to start by taking a look at where medicine has done phenomenally well versus where the gaps are. And as I said, many people have looked at the hallmarks of aging, said it's a phenomenal model, but where to next? So let's just look at medicine for a second. And if you look at the diseases that it's been immensely successful at intervening in, these are diseases you see early in life. For instance, your congenital defects or your infectious diseases. And typically, when you look at these diseases, what you see is a single underlying driver, right? So if you think of an infectious pathogen, that may be a virus, a bacteria, a parasite, or so on. And targeting those diseases has been far more straightforward because you can then develop a single drug for a single target to intervene the single, underlying problem within that disease, and therefore develop a drug for a single disease, right? So it's a single drug, single target for a single disease. Now, the complexity of aging is such that this approach just does not work for twofold reasons.
Mm-hmm.
The first is many diseases are multifactorial. And all that means is the same disease can be driven by multiple different factors, different underlying causes, right? So again, if you picture, what you hear later in life, if you're going into a doctor's office, you hear things like high fat diet increases risk or smoking increases risk. And you get these multiple risks because these diseases are so complex and they are multifactorial. The other side is multimorbidity. And all that word means is multiple different diseases coming on simultaneously with no one clear underlying cause. So you may see cardiovascular disease alongside kidney disease, alongside dementia, the list goes on. So the aim with this blueprint theory was very much, let's appreciate the fact that aging is a systems biology problem.
Mm-hmm.
And therefore, let's take a systems level solution to this problem. So that solution has been akin to what you see in finance with complex systems, which is factor modeling, where in a very complex system, can you identify key nodes or factors driving the maximum level of negative change and intervene in those factors such that you are going to have large protective effects across the system as a whole.
Mm-hmm.
And really trying to identify those nodes is how we landed on necrosis.
Mm-hmm.
So I just want to set the scene there. Okay, so now delving in, what is necrosis and how is it different from the programmed cell death forms that you have? So programmed cell death simply means that they are genetic programs that have been selected for that give rise to these forms of cell death, like apoptosis.
Mm-hmm.
And evolution has selected these forms of cell death because they confer large protective beneficial effects, such as wound healing, developmental changes, tissue remodeling. So if you get a fracture and you need to heal the fracture, you need this form of cell death to remove the bad material before you can build a new. And of course, removal of cancer cells. So in contrast, necrosis is cell death that arises from cellular damage. It is not governed by any genetic programs, and it's just a systems level collapse of the cell. And so it's rather interesting if you think of your physiology as a whole, what are these key nodes? Necrosis is a key node that underlies tissue degeneration across the system. And then, of course, at the cellular level, again, it's system level collapse of that cellular system that is necrosis.
Mm-hmm. Mm-hmm. And you brought up your your blueprint theory, which you outline really nicely
Mhm.
also in this recent British Journal of Clinical Pharmacology that was just out in October a couple weeks ago, called Beyond GLP-1s. But there's a really interesting paper a couple months before that you put out connected to this, and
Mhm.
this was in Nature Oncogene in July. It was a review you wrote entitled "Necrosis is a Fundamental Driver of loss of resilience and biological decline." And this is fascinating because as you're just talking about these, these pathways and nodes, it's in this paper that you really talk that you basically point out necrosis is more than just an endpoint, right? I mean, you got cell death, but what follows? I mean, there are these cascades of feedback loops that lead to more necrosis that ultimately trickle down to other organ systems that destroy, let's say, this foundation of resilience. And I wonder if you could just spend a little time on this, because again, the principles that you're pointing out, again, as you were just talking about the patho-pathways in the recent paper, it's very different than, when we think about upstream, rapamycin and metformin and all that stuff and the Gerotherapeutics. You're doing something a little, I mean, your nodes are different. I mean, they're sort of upstream, but they control much different stuff. And that's why I wanted to sort of discriminate here between what we normally hear about in terms of traditional gerotherapeutics and, okay, we're hitting AMPK1 or whatever. You're impacting something a little more, complex. And I want you really to delve into this because I think it's extremely important with where you're going in your drug development.
Sure. You hit the nail on the head there, which is to date, not a single drug has been approved for aging. You hear many of the drugs like metformin, rapamycin, and so on. But the truth of the matter is none of these have been approved by any of the major regulators. Not the FDA, not the European equivalent, the EMA, UK equivalent, MHRA, and so on. So why is that? And for me, this radically different approach came from trying to get the first drug approved for aging.
Mm-hmm.
So, what are the challenges that you typically see? And the key one is the lack of mechanistic information, clear mechanisms, and clear link to indications that the regulators want. How is a particular mechanism from your molecular level driving your cellular changes, your tissue changes, to finally cause some sort of age-related decline that you can see at a tangible, physiology level scale? And that could be your life-limiting diseases, right? Your cancer, Parkinson's, Alzheimer's, or the non-life limiting pathological side of aging, your skin wrinkling, hair going gray. I still refer to these as pathological because, they have unwanted negative consequences, right? Is just one example. If you lose your skin barrier function, you have skin related changes, it means there's a higher risk of infection or extrinsic insults such as toxins and having a negative effect on the body. But right, so how do you take this fundamentally different approach to all of these other drugs? And how do you get those mechanisms? And the answer there is to start to map out all of the different changes, right? As I mentioned, I'm not the only person who said we need to dig deeper when it comes to the hallmarks of aging, or there's some missing gaps. But what is novel here is identifying the solution to the problem. So how do you start to put the different hallmarks of aging into context to understand your molecular changes to your cellular changes to your medical disease? And really, what that paper pinpointed is when you start to map out the different changes, necrosis really stands out as a key node or a key, if you like, a key point within the chain of events through which multiple different changes transmit, right? If you think of it as a grid, it's a key node within this grid. And let me bring that to life for you. So necrosis occurs when you have cellular damage. So if you have a stressor on your cell, what happens is you have internal cellular damage taking place, and that takes several different forms. That can include cellular damage causing genomic instability, hallmark of aging, loss of all of your protein regulation or loss of protein homeostasis, another hallmark of aging, mitochondrial dysfunction, and so on. So it's the same cellular damage that causes these. So at lower levels, what happens is it can cause just these sort of dysfunctional states within the cell, the same pathway, the same necrosis pathway of cellular damage. At harsher levels, it results in cell death through necrosis, unprogrammed cell death, right? So when the stress is harsh enough, you don't just get internal cellular dysfunction, you actually get the death of the cell. And when that happens, it's a very messy process because in effect, your cell ruptures, it pops within the tissue, it spews out all of this intracellular contents, and then it triggers further cascades of necrosis because it triggers further damage. But what it also triggers, because you're spewing out all of these hazardous contents, is dysfunction within the tissue in the form of chronic, persistent, difficult to treat inflammation, right? So again, for the hallmarks, fibrosis or scar tissue formation, and also it can trigger senescent cell accumulation. So again, where is the senescent cell accumulation coming from? It's starting to put together your causal chain of events. And you can see this role of necrosis across the whole system. You can also see it within individual organs within the system. So for me, what was key here is A, the fact that you're able to map out the mechanisms, right? So the one missing element to gain that regulatory approval. But B, once you map it out, the fact that necrosis is such a key element, where if you were able to stop the cellular damage, you would be able to stop so many of these hallmarks of aging. You would be able to stop this, if you like, the chain of events that's then driving, or the bulk of the chain of events that's driving your tissue degeneration.
So again, just to sum up for the audience, so we have this necrosis, which is clearly a patho-pathway, as you point out. If we can inhibit it, we can prevent various forms of cell death cascades, and ultimately, you know, that again, thinking it as a separate thing from the sort of the metabolic reprogramming of the metformins and the rapamycins. This is really damage control pharmacology, putting out these fires before they become worse fires. And so, yeah, I mean, it's a beautiful and very elegant set of thinking here. Now, with that, I know I would love to go into sort of LinkGevity and a little bit about how you've sort of preclinical sort of focused in. I mean, I know your lead asset is something known as Link-001. It's going to be focused on acute kidney damage. Could you talk just, I know I tried to take a look around sort of the IP area, and I saw some sort of patent applications on ryanodine-receptor inhibitors and calcium chelators and some repurposing. But whatever you can talk about, Carina, in terms of, you know, how you focused in, maybe using some of the AI on what made, in this particular case, this perfect first generation necrosis inhibitor for the company, and a little bit of sort of mechanistically what it's doing, that'd be cool. Whatever you can talk about.
Of course, happy to. Just one key element with the necrosis I have to mention is the key to this is you see these vicious cascades of necrosis disrupt your tissue and cause your degeneration. If you were able to intervene, the key is you would get natural regeneration because you would resume natural cell division. And I think that's a very crucial point and a crucial value of this technology. But yeah, of course, I am happy to go into the mechanisms around necrosis. So, for a long time, it was actually assumed that it's near impossible to intervene in necrosis because it is unprogrammed cell death. There are no genetic programs, and it is so chaotic, right? Just think of all of your forms of cellular damage. You're having structural damage, you're having dysfunction at different organelles, you're having molecular level dysfunction, metabolic dysfunction. How on earth do you even intervene? And again, it goes back to this principle of factor modeling. Yes, it is a complex system. Yes, there is some chaos within that system. But can you pinpoint the right nodes such that if you were intervening in those nodes, you would have the maximum protective effect across the system as a whole, right? If that principle makes sense. So you're not taking out everything, but you're taking it out enough to have a 90 to 100% beneficial outcome. Okay, so what is that at the molecular level? And this really goes back to work in the 1970s, where it was identified that the first thing to fail at the molecular level within cells is loss of calcium ion gradients. So, just for those who don't have an in-depth biological background, typically you have different ions like calcium, sodium, potassium, and so on. And they're at different levels outside the cell versus inside the cell. And so, one of the first things to fail upon cellular stress is all of these different gradients, these different levels of these different ions get skewed. Why is calcium particularly so disastrous if those gradients get skewed? Well, the answer there is calcium is a key signaling molecule. In other words, levels of calcium within the cell determine functions that arise within the cell. And typically, levels of calcium outside the cell are 10,000 to 100,000 times higher within the cell. Just try and picture that difference, right? And so upon stress, you get something known as calcium overload. Calcium floods the inside of a cell. When that happens, it switches on multiple signaling pathways within the cell simultaneously. In other words, multiple functions are switched on, only to a heightened and destructive level. And that then gives rise to this chaos of loss of protein homeostasis, your DNA damage, your mitochondrial dysfunction, and so on. In fact, this is why necrotic lesions are at such high risk of cancers, because you have this ongoing DNA damage. But the point is, the key node there is this loss of calcium ion gradients. And to date, there have been numerous attempts to try and block that, albeit unsuccessfully. What we managed to do with the factor modeling is actually identify the correct set of molecular targets to hit simultaneously. And so for the first time, we were actually able to block necrosis. And the data was actually so striking. In effect, what the drugs can do, what this anti-necrotic drug can do is shield cells from stress. So you can take cells, you can expose them to some of the harshest stressors. So remove, really reduce their oxygen, for instance, or even put them into hydrogen peroxide or expose them to high levels of heat or cold. And typically, where 100% of those cells die, with this anti-necrotic drug, 90 to 100% of those cells is robustly still alive. And in fact, the data we were getting surprised us so much, we had to take it to independent organizations. So they're called contract research organizations that can independently perform experiments and have them validate the data. But long story short, that work was done. That was all part of the preclinical development. We were careful to use human cells wherever possible, because you want to make sure this translates to humans. And in fact, across the board, across cell types, we're seeing the protective effect. And yes, of course, what I can go into later is in terms of those studies, once we looked across the human body, we then decided to focus in on the kidney. Key reason for that is simply because the kidney shows accelerated aging. In other words, you can do the trials in a much shorter time frame.
Yeah. Yeah, let's talk a bit about that because I have uh, I had a little experience in chronic kidney disease, and I remember how long those studies had to run. But you've, just strategically for the company focusing on acute kidney injury, which the trials can, be quite a bit shorter. Could you talk a little bit about just again, and you're very elegantly talking about, okay, we have this nasty calcium event that's happening, and you're going to be intervening potentially at different pharmacological interception points of calcium release and downstream effectors of the calcium in cells and all that, which is awesome. It may bring us over to acute kidney damage. What type of insults, damage, infections, whatever, you're going to be looking at in terms of, okay, this is an initial first set of indications here, and a little bit of how you think about what this looks like clinically, for us, please.
Yeah, so to date, kidney disease, it's the ninth leading cause of death, as per the World Health Organization. It's also an unmet medical need, because to date, no drug can directly intervene in kidney disease. You only have treatments that mitigate it. For instance, you have dialysis or transplants, neither of which are particularly pleasant for the patient. Mortality rates are really high on dialysis, not to mention quality of life is reduced. And with transplants, it's quite frightening. 80% of patients typically die on the organ waiting list, waiting for a kidney.
Mm-hmm.
So, not the most pleasant disease, and it's an unmet medical need. And if you ask yourself, why has it been so difficult to intervene in kidney disease? The answer goes back to it being a multifactorial disease. Multiple different factors are giving rise to kidney disease. And this includes changes in blood pressure, a lack of oxygen and nutrients, or ischemia, right, hitting the kidney, inflammation, side effects of some of the drugs one takes, such as antibiotics or pain medication. And so
Mm-hmm.
all of these can impact the kidney. And all of these condense onto one particular pathological process, which is acute tubular necrosis. So
Okay.
that is necrosis of the cells doing the bulk of the filtration within the kidney.
Mm-hmm.
Now, why is the kidney in particular so vulnerable to stress? And the answer is for twofold reasons. The first is it's one of the most metabolically active organs in the human body. And typically, we think of the brain as one of the most metabolically active. The kidney is equally, if not more, metabolically active when you account for mass. So, when you normalize to actual mass of the tissue, because of course it's performing all of the filtration of the body. At the same time, it consists of less than 1% of the body mass, and yet takes 20% of your cardiac output. Right? So very vulnerable. Hence, well, arguably, hence evolution came up with two kidneys.
Mm-hmm.
So, question here, do we know the mechanisms in terms of what happens when you have these different stresses hitting the kidney? The chain of events, the chain of events that includes the different hallmarks of aging, if you like, and then your end outcome of kidney disease, your chronic kidney disease. And the answer is these have been heavily mapped out. And typically, what happens is once you get your acute tubular necrosis, so necrosis of those tubule cells, it's the same chain of events I mentioned earlier. You get this vicious feedback loop where necrosis then propagates. You get these cascades of necrosis taking place. But also, what you see in the kidney is that necrosis then drives this chronic, persistent, difficult to treat inflammation, scar tissue formation or fibrosis within the
Mm-hmm.
kidney. And it also triggers senescent cell accumulation. So, your hallmarks of aging. And it is those that then give rise from your kidney injury to its progression to your chronic kidney disease.
Mm-hmm.
So you can start to put together the chain of events, right, in terms of the aging phenotype, but then also the end outcome of the disease. So what we want to do with this intervention is really tackle the root cause, tackle the acute tubular necrosis, and therefore tackle it at a stage before it progresses down through the aging phenotype and then also to the end stage of kidney disease. So that is the initial trial. That being said, of course, there's also scope for a treatment element here because, as I said, if you can these vicious positive feedback loops of necrosis at their heart, what you would try and allow for is natural cell division to resume a natural, if you like, physiology of that kidney to reestablish.
Yeah, that's a beautiful two-in-one opportunity with what you're doing. And again, we forget about the sort of the regenerative potential of many of these systems. We think, you know, they're kind of set in stone, but yeah, I mean, what you're pointing out is extremely sort of crucial separate piece of that. But and again, as you're saying, the endpoints in studying acute kidney injury are quite different than what you have to study chronically, and so you'll be able to get a readout much faster, which is exciting. What, you know, in addition to you know, calcium, and I know this, you know, initial agent is focused on that. Could you say a couple of words about, because you know, in sort of preparing to talk to you today, I was sort of reading up on some of these other forms of necrosis that we know about now in terms of ferroptosis, which is more sort of iron dependent, and something called pyroptosis, which has its own. I mean, do you envision sort of a whole suite of necrosis inhibitors that, you know, are you modify based on your system to, you know, because they're, you know, as you pointed out in your earlier paper in Senescence, there's all sorts of necrosis also. So talk a little bit about your strategy with, you know, what happens with some of these other forms of necrosis that are not strictly calcium dependent, and where you may be going with that with the company.
So this is covered actually in that Nature Oncogene paper. The fact that really you have two types of cell death. And by the way, I picked that, you can pick that particular journal because always whenever you think about cell death, you're thinking about cancer, right? That's the first thing at the back of one's mind. If you start playing around with cell death, are you going to play around with cancer?
Right.
But broadly, again, you have two types of cell death. You have forms of cell death that are genetically programmed, and actually all of the forms you mentioned fit into that. The necroptosis, pyroptosis, ferroptosis, apoptosis, the list goes on. So all of those fit into the genetically programmed bucket of cell death. In contrast, necrosis is purely unprogrammed cell death. So there are no genetic programs underlying necrosis. You're absolutely right that there are certain links in terms of presentation of different forms of cell death. And just to delve into the science here in a bit detail, because you need this, the question is, with the programmed forms of cell death, when you see those forms of cell death, right, your ferroptosis, necroptosis, and so on, where are they coming from? Because typically they're always good, right? Evolution has, or if you like, via evolution, we've focused a means to have these forms of cell death because they confer some sort of a benefit. So why do they arise? And this goes back again to your cellular damage. So this calcium-driven dysfunction within the cell, this necrotic pathway. So that pathway can be initiated to varying degrees based on how much stress the cell has been exposed to. And you can really picture that along a continuum. But across that continuum, you have three broad buckets. The first bucket is where it is so harsh that unprogrammed cell death necrosis is the outcome. The cell just ruptures and spews its intracellular contents. The second bucket is where that same necrosis pathway, the same pathway of unprogrammed cellular damage, causes enough intracellular damage not to directly destroy the cell, but enough damage to then be picked up by programmed cell death pathways. Because then your cell appreciates the fact that it is damaged and it should stop itself from dividing before it propagates into cancer.
Mm-hmm.
And typically in your medical textbooks, whenever your tissue is damaged, you see a lot of necrosis. You then also see mixtures of these programmed forms of cell death because they've come about to try and get rid of those damaged cells. In fact, you hear words like panoptosis because you have simultaneous pyroptosis, necroptosis, ferroptosis.
Mm-hmm.
But if you ask yourself where they're coming from, it's because they're getting rid of the cellular damage.
Yep.
And of course, the third bucket is you have damage not enough to kill a cell, directly or indirectly, but enough just to cause internal dysfunction within the cell,
Mm-hmm.
right? So you can then trigger things like cell senescence and so on. And the crux of the technology is you do not want to stop genetic programs associated with beneficial cell death, because
Right.
if you do that, you're going to risk cancer.
Right.
But what you do want to do is get rid of the cellular damage upstream. In other words, intervene in that necrosis pathway before it gives rise to either of these three buckets.
Mm-hmm.
And that's what the anti-necrotic is really geared at doing.
Got it. Um, Really fascinating. Um, Karina, let's go a couple of months prior to the resilience paper to experimental physiology, March 2025. Beyond the horizon, why space biology is the next great innovation and opportunity. And over the last couple of years, we've hosted folks like Dr. Matthew Roberts, who's Chief Science Officer at the International Space Station National Laboratory here, also Colonel Sam Weeks, who worked on the SpaceX Polaris Dawn program, and they had a whole radiation biology initiative there. You point out some really interesting things here, and that basically space is quite dangerous. A lot goes wrong. It's not just about zero gravity or radiation or a million other things happen up there. And clearly, everything sort of converges here on necrosis, and as you point out in this paper, kidney deterioration. And it's one that we hear about, sort of the people on the space station. They got eye problems and bone loss and issues with the heart, but we don't really hear as much about the kidney. And you point out that, look, this is probably a very important target, whether we're headed back to the moon base or Mars or wherever we're going. And I would love to hear just a little bit about some of your work here, because I know this is a very important part of your research. But at the same point, and Mr. Musk or whoever wants to get us off this planet, we really got to think about necrosis in space. So take us down that path.
Space is a really harsh environment for the human body, as you can imagine. What I always say is problems on Earth are problems in space, only magnified. And so the same challenges that give rise to your degeneration and, age-related changes on Earth are compounded in space by microgravity and cosmic radiation. And so what you end up with is really accelerated tissue degeneration and early onset of many age-related changes that you would see on Earth. If you think back to earlier this year, the astronauts that were on the International Space Station that were stuck there for 9 months, why was that so frightening? For them, it is because of this rapid degeneration that one can get. And remember, this is still when you're within low Earth orbit. And so you're within the shielding of the Earth. Imagine when you were moving beyond that to long duration missions. And again, yes, you get degeneration across the system, but the kidney is really one of the most vulnerable points. It's an organ that demonstrates accelerated age-related decline and also degeneration. And that's not just true on Earth, it's also true in space. In fact, one of our collaborators led a large study alongside NASA, the European Space Agency, also the Japanese Space Agency. Interestingly, also had data from the Chinese Space Agency. So one of the most international studies on what would happen to an organ in space. And they actually focused this on the kidney. Again, owing to the fact that it is so susceptible to these changes. And long story, it's spoiler alert here, but long story short, the end outcome of that study was, would astronauts even survive a trip to Mars, or would they need dialysis and transplants before they even
Right.
get there?
Yeah. Yeah, that's I'm glad to see you're working on this and thinking about it, because clearly this is, I think, an underappreciated area of, again, getting us into space again on a large scale and spacefaring society. But yeah, it's a quite interesting topic, and I see why it fits so nicely with, again, how you think of this type of pathology and these different patho-pathways. Um,
Yeah.
one other thing, please.
No, I
Please.
was just gonna say, absolutely. Look, I see this as a big hurdle, right? Whenever you
Yeah.
think of space, you always think of the hardware, the satellites and rockets.
Yeah.
Perhaps the biggest hurdle is going to be our own biology. But the only thing I was going to add is it's also a large opportunity. And just because you mentioned that particular paper, the paper goes into this. I think space is also a very large opportunity to rapidly develop biological interventions. It's one of the things we're looking at,
Hmm.
scaling technologies. And if you think back to what space did for, your engineering field, from the mundane to the big leaps, from,
Hmm.
something as simple and common as your scratch-proof glass to your microchips.
Hmm.
The question is, can you use space as a laboratory to try and accelerate development of technologies for biological challenges we have on Earth? Could space do for biology, what it did earlier for engineering?
Hmm. Absolutely. Absolutely. And talking about laboratories and space being a great one, I would just really appreciate, because you at the same, well, this is in April 2025 in Nature Medicine. This is a piece that you wrote on the human exposome with the whole gang of a lot of these folks I've had on the show: Near, Brian, Jamie Justice. Um, can you talk a few minutes just about where we are in terms of understanding human exposome knowledge, and specifically how it relates to your area? Because whether we're talking about chemicals or physical processes, or what, biologic threats and so forth, a lot of this converges ultimately on necrotic events. And I'd love to, again, thinking about laboratories in interesting ways. One place is space, but this whole principle of the exposome is really fascinating. I'd love to just get a few minutes from you on, so your aspect, well, your connection to this paper is obviously very important, but a little bit of your thoughts in the sort of the exposome necrosis link.
Yeah, so the exposome, again, just some background for anyone who hasn't come across this. So the exposome is very much trying to understand how environmental interactions can affect our health. Right. And this goes back to the idea we had a few decades earlier, which is perhaps all of health decline and all of these degenerative changes with aging are somehow linked to our genomes. And then you had the Human Genome Project. And the point was, when we finally complete the Human Genome Project, we would have solved aging. And the point is that's been completed and we haven't solved it. And we haven't, because there's this whole other layer that one needs to now unravel, which is these environmental interactions.
Yeah.
And I think that's often been not appreciated enough. Um, because if you look at some of the more recent studies, you may think, okay, it's predominantly your genome that dictates, your aging trajectory. That's not true. The environment plays a very large role. So some recent examples, there was a paper that looked at 22 major diseases associated with aging. And they looked at environmental versus genetic outcomes.
Mm-hmm.
And interestingly, they found if you look at the variation, only 2% could be explained by genetic predisposition, 17% could be explained by environmental factors.
Mm-hmm. Mm-hmm.
So the key here is the environment plays a very large role in determining your trajectory, your aging trajectory, hence the space as well, right? Why does space accelerate aging? Because the environment plays such a large role. So the
Mm-hmm.
point is, how do you begin to disentangle all of this complexity? The Human Genome Project itself took so many long years, global collaborators. Imagine that, but now on top of it, you have to try and account for all of these different environmental variables. So I think the key there is, when we started looking at it, again, I'm going to go back to the factor modeling. That is the approach we took. And we asked ourselves, what are the key factors, not just associated with the genome, but with these environmental interactions? And yes, necrosis does come up with a key interface as well between environmental stressors and how you drive then internal degenerative change, right? The cell is the fundamental unit of biology. It's your fundamental building block. And how that cell lives or dies then determines the health of the organism ultimately.
Mm-hmm. Yeah, no, it's again, it's another really fascinating piece of your work, and the connection is kind of self-evident to where you're going with the company, but it's yeah, it's really impressive. What else should we be on the lookout for in terms of you, the company in 2026? I mean, just looking around, I mean, I know you're going to be in Tokyo next May at the International Conference Asian Gerontology. You're at SymBioBeta. You're at a couple of weeks. You're at SpaceCom in Glasgow. It seems like you're really getting out there and spreading the word about everything that LinkGevity is doing. What else is happening that we should know about that's public facing? And then anything that we missed that's coming up in 2026 that you want to mention while we have you today, please.
No, you've covered everything. For me, it's always science first. Science drives everything that we do. And the next stage for us is subject to regulatory approval, starting our clinical trials. Very ambitious trials. The aim will be to really get the first drug approved, yes, hopefully for kidney disease, but also the bigger side to this, use the kidney as a model to try and, gain regulatory approval for aging as an indication. Again, because you have that unique position of the mechanisms having been highly characterized. The other side is we're also looking to take the anti-necrotic into space. So we've
Mm-hmm.
actually received funding from the UK Space Agency,
Nice.
take it up, study its structure, and so on. That's going to be happening in parallel. And I guess really for me, maximizing impact of what we already have. So with the anti-necrotic, something I haven't really gone into is yes, necrosis has been a fundamental challenge when it comes to medicine, but it has also been a fundamental challenge in scientific research and development.
Yeah.
And just to give you one example there, in tissue engineering, so creation of artificial organs, for instance, in a lab,
Yeah.
one of the key hurdles is you cannot grow these artificial constructs to scale because the central region gets starved of oxygen and nutrients, and you end up with a central dead core or a necrotic core. And the challenge has always been, can you grow your blood vessels, your vasculature quickly enough before that core forms? So again, because you're able to block necrosis and for, such an extended period of time, you have applications in many other areas, like in tissue engineering, which could enable for the first time growing far more complex artificial tissues. So the other side for us is very much ensuring that we maximize the impact of this technology and find the right partners to do that. So that's the other
Mhm.
side.
Yeah, Aaron, I made a mistake in not mentioning that as your this non-clinical pipeline. You have we'll link to it in the show notes, and again the connection. I mean, we've had all the sort of the godfathers and godmothers of organ engineering on the show talking about this oxygenation issue, but the connection not just to tissue engineering, but another project is Link 003 and organ preservation, and small small changes to necrotic processes there. That's a major holy grail as well. So I'm excited to see you working in that space too, which is also very important. But I love what you're doing. I'm excited for you. I look forward to continue to follow you and the company as you get into 2026 and get the first asset into clinical studies. But really impressive set of work, very different than a lot of the aging themes that we touched on in the past. So I'm looking forward to what comes next. Again, for everybody out there that is going to be listening to this episode on the various podcasts, or who will be watching with us on the YouTube channel. Again, you've been spending time with Dr. Carina Kern, Chief Executive Officer, LinkGevity, doing really amazing work at the intersection of drug discovery, aging, loss of biologic resilience, and creating really what we need, translational geroscience opportunities. Carina, again, I want to thank you for taking the time to talk to us, to walk us through everything you're involved in there. Obviously, thank you for everything you're going to be doing for patients. And as we like to say here on our show, thanks for helping to create a better tomorrow for so many people via what you're trying to champion there. Really an awesome story.
Thank you. It was an absolute delight.
Great having you.